Today we are going to explore something very ordinary in an extraordinary way. This is a tomato. We eat it every day in our salads, curries, and sandwiches. But have you ever wondered what a tomato looks like under a microscope? From the outside it looks smooth, shiny, and red, but inside, this simple fruit is an entire microscopic world waiting to be discovered. Now it's cut open and let's see what's inside. As you can see, the inside is juicy, soft, and filled with seeds. All this red pulp is made of thousands and thousands of plant cells. I'm taking a very small piece from the inner pulp, the soft red part. For microscope observation, we need a very thin and small sample. I'm using the forceps to carefully place this tiny piece onto a clean glass slide. Now I'm adding a drop of the tomato juice which I got it from this tiny piece. This helps spread the cells evenly and prevents air bubbles. I'll gently spread it a little so that light can pass through it properly. In microscopy, thinner samples gives clearer images. Now comes an important step, placing the cover slip. I'm gently lowering the cover slip at an angle by reducing air bubbles and protecting the specimen. And now our tomato slide is ready for microscope observation. What you're seeing here is a fresh wet smear of the inner pulp of the tomato. I did not prepare a thin tissue section. This is simply the natural sap placed directly onto the slide. Throughout the sample, the view looks almost the same, scattered round particles, uneven clusters, and a slightly cloudy background. Those numerous small, dark, round bodies you see are most likely chloroplasts. Chromoplasts. These are pigment-containing structures that store lycopene, the compound responsible for the red color of the tomatoes. Since tomato fruit tissue is very soft and made of loosely arranged parenchyma cells, many cells naturally release their contents when cut. As a result, instead of seeing neat rectangular plant cells, we are mostly observing suspended chromoplasts and cell sap. The large circular shapes visible in some areas may be intact cells, partially collapsed cells, or even small air bubbles trapped under the cover slip. Because this is a sap smear and not a structured peel preparation, the pattern remains similar across the entire field of view. Dispersed pigment bodies floating with the liquid matrix. This gives us a microscopic look at the internal pigment system of the tomato rather than its organized cellular structure. If you enjoyed exploring the microscopic world of tomato, stay tuned for more real-life samples under the microscope. See you in the next experiment.